This video is over chapter 17. We'll be talking about regulation of gene expression in eukaryotes and how that differs from gene expression in prokaryotes. So as we mentioned before when we talked about translation, gene regulation in eukaryotes is going to be a lot more complex than in prokaryotes, mostly because in prokaryotes, the DNA is contained in the cytoplasm, whereas in eukaryotic cells, that DNA is contained in the nucleus, and it's much larger. It's linear. It has an association with histones and other proteins. And so most of our genes that then get turned into mRNAs, they need to be spliced, they need to be capped, they need to have the poly A tail put on that, and then as they exit the nucleus and move into the cytoplasm to be transcribed, will be transcribed by the ribosome and then translated. And so all of this necessitates more points at which we can have regulation of gene expression. To begin with, let's talk about the actual chromosome itself and what we call chromatin modifications. So chromosomes inside the nucleus, they actually occupy a very discrete territory. They stay in those areas and they're actually separated from other chromosomes. Even though it looks like it's just a big mess of nucleic acid, we know that transcription and translation are spatially distinct. Now, that eukaryotic DNA is combined with proteins that are called histones. There's additional non-histone proteins that are also associated with the DNA, and that's what forms what we call our chromatin. And chromatin, it inhibits transcription, replication, and even DNA repair. It doesn't allow the enzymes associated with those processes to be involved. And so before we can transcribe, replicate, or even repair DNA, we have to move those histones and other proteins out of their way. So during interphase, our chromosomes are found in their discrete chromosomal territory, and there's small channels between those territories that are called interchromosomal domains. And it's in these domains that we then have what we call transcription factories. So these transcription factories are the sites that contain the most active RNA polymerases, and this is where transcription and regulatory molecules will be housed. Now, in order for us to utilize and access these regions of the gene, generally we have to go through what we call chromatin remodeling. This is a really important step in gene regulation because if we alter our chromatin, we can either turn on or turn off genes. And when I say turn on and turn off, what I really mean is make genes accessible or inaccessible to these transcription factories. And we can do chromatin remodeling in one of two ways. One is we can either make changes to the nucleosome, that is the histones and proteins associated with those histones, or we can make changes to the DNA itself. Let's begin first with nucleosome alteration. So here, there's three mechanisms by which we can alter the chromatin itself. The first is what we call histone variation. So most of our nucleosomes, and again, the nucleosomes are histine proteins in which we have our DNA coiled around. So those nucleosomes generally contain either H2A or H3 histones. There are, however, variants of those histones, and they can help facilitate gene transcriptions. And these variants, the way they can, if we use, say, like an H2AZ or an H3.3 histone, that protein structure allows the promoter regions to be free. And so you can have normal repressive nucleosomes out of the way so we can transcribe those sections of the gene. So the analogy that I like to give for these nucleosome alterations, imagine, if you will, that you are a parent and you and your spouse are walking down the street with your toddler. Okay, the toddler in this scenario represents the gene. Okay, the parents represent the nucleosome. So you're tightly bound to your toddler because you're protecting that toddler. Now, parents come in different shapes and sizes, right? Some parents may be helicopter parents in which they do not let that child out of their sight and are constantly holding the hand. Maybe they're the parents that have the leash on the child that's attached to the little monkey backpack or whatever. And they do not let strangers come anywhere near their child. Strangers being transcription factors, right? In some cases, even helpful strangers, like say a teacher or something else. They just don't want them around. Likewise, you have different parents that may be way more laid back. They may take an attitude of, well, let's let our kid have interactions, and that'll be all right. They kind of hang back and allow transcription factors to access the gene. Okay, just switching out the type of parent, or in this case, the type of histone that you have, can allows for that type of interaction. And the way it does that is the DNA becomes perhaps more loosely wound around those nucleosomes. So that's the first, histone variation. The second mechanism is histone modification. So in this case, we're not changing our histones. We have the same histone. It could be an H2A histone. But this time, we modify it generally through acetylation that causes the histone to have a less of an attraction to the DNA. So our DNA histone attraction becomes less. The way we do this is we use what we call acetyltransferase enzymes, or HATs. And these HATs add an acetate group onto the polyase onto the amino acids of our histones. And so now these amino acids have an acetate group on there, and that tends to make them less likely to be attached to the DNA. We also have histone deacetylases that remove the acetyl groups from those histone tails, that makes them even more attached to the DNA or attracted to the DNA. And these can occur through either phosphorylation or methylation. We'll talk about that in a minute. So going back to our analogy of the parents and the child, the parents being the histones, the child being the promoter region of a gene, you can talk about histone modification would be like if you put the parents on chemical substances. So if the parents were to drink a bunch of alcohol as a depressant, that would be like acetylation, right? They're going to become a little bit less, they're not going to be as active. They're not going to be keeping an eye on the child. They're probably not going to hold the hand. Take it a step further. Let's give them a really big depressive substance. Let's say they take a hit of an opioid, right? Of heroin. They're going to basically be asleep, right? They're not even paying attention to what that kid is doing. That would be histone acetylation. The deacetylation would be like the opposite. Let's say you give them cocaine, right? A stimulant to where they're hyperactive. That normal helicopter parent now becomes a hyperactive helicopter parent. Nothing is getting near that promoter region. Okay, so we've had histone variation, histone modification. The last is what we call chromatin remodeling. And in this one, now we're not changing the histones themselves, we're not modifying them, but we're utilizing additional complexes called chromatin remodelers to reposition, or in some cases even remove out the nucleosomes of the DNA. So you can see right here, this is an example of our SYI, SNF chromatin remodeler. We understand that protein pretty well, utilizes energy, and one of three things kind of happens, right? We can either loosen the attachment between the histones. So here's two histone groups that are pretty well bound to each other. But using this chromatin remodeler, we can actually slide them apart, and we have this exposed region. We can have alteration of the DNA path. And so instead of being wound, in this case, rather tightly, we can pull some of this off, and now it's more accessible. So that loosens the DNA from it. And then we can also reorganize the nucleosome. So we can take these histones and put them into two dimer forms, which again, we have more of that DNA that's accessible. Okay, so this is using a intermediary. So again, going back to our example of the parents that act as the histones or nucleosomes, and the child that acts as the gene promoter. Now it's where you have somebody come in and literally get and pull the parents apart, reposition them. So maybe you'll have a social worker or a friend or somebody says, hey, stop hovering, give this kid, let him breathe, move around. Or maybe you have somebody that does the opposite, that brings them in and says, hey, you got to get closer, you got to be careful. There's a bunch of dangerous strangers in this area, so bind yourselves a little bit tighter. Okay, that's chromatin remodeling, a third-party group kind of either pulling the parents apart, pushing them together. That's all the three ways that we can alter DNA regulation by just messing with the histones and the nucleosomes. But we can also address the DNA itself. And one of the largest ways that we do this is through DNA methylation. So in DNA methylation, what we do is we tend to methylate a cytosine. So on the 5' position of the cytosine, we add on a methyl group. And when we do this, what we find is that we have a decreased gene expression. And so there's areas in our genome that tend to have a lot of what we call CG repeats. So we go CG, it has a high cytosine guanine content. And in these regions, we tend to get these doublets that get methylated. We call them CPG islands. And these CPG islands tend to be found towards the 5' ends of the genes, which conveniently are in the promoter regions. And at any time in our genome, about 5% of your cytosines are actually methylated. And we know that methylation decreases gene expression. By having these methyl groups on there, it changes both the affinity that, say, histones may have to that DNA, as well as the ability of transcription factors and polymerases to bind on. This has been really well studied. There's lots of evidence of this. For instance, there's an inverse relationship between methylation and expression. We see that in humans with our X chromosomes. The X chromosome that's inactivated is very, very highly methylated. Similarly, we see this in tissue-specific patterns. So we can have methylation patterns that tend to be propagated in daughter cells from those tissues. So all the cells inherit that, and we can see that there's lower expression in those tissues than, say, in other tissues. There's also analog studies in which we basically engineer methylation. We use a nucleoside 5-azacitidine substitute for our cytidine during replication. And this cytosine substitute can't be methylated. And when we put this in to organisms that then are replicating themselves, we can actually take, like, for instance, in mice, we can stimulate the expression of alleles on that inactivated X chromosome. So, hypotheses of how methylation might work. Well, it's either inhibiting the binding of transcription factors to these regions, or it's recruiting repressive chromatin remodeling complexes to basically make those areas inaccessible. Both of those are valid hypotheses that we seem to find some support for. All right, so we've talked about our chromatin and our DNA. So how we can, how the eukaryotes can regulate whether genes are accessible or inaccessible by, nucleosome and histone complexes, remodeling and variation, as well as effects to DNA itself, like methylation. Now let's move on to how eukaryotic transcription itself. So now we're not talking about just the ability to start it, but now transcription, how we regulate that. And we need to go over some vocabulary words for this. You'll remember that promoters, those are cis-acting sites. They're on the same strand as the gene itself. And generally, transcription is going to require the binding of many regulatory factors to these cis factors. So we're going to have lots of transacting elements binding to these cis-acting sequences. And the sequences that we have promoters, enhancers, and silencers. We're going to talk about those first. So first off, what is a promoter? We've used this concept many times before. I don't know that we've actually defined it, but a promoter is a nucleotide sequence that serves as the recognition site for our transcription machinery, meaning transcription factors, RNA polymerase, etc. Okay, it's critical for the initiation of transcription. And it's generally going to be located adjacent to the genes that it itself will regulate. We can divide up promoters into two types. We have what are called core promoters and proximal promoters. So core promoters are where our RNA polymerase II specifically binds. I cited this is a typo. This is the regions that determine the accurate initiation of transcription by our polymerase. So polymerase binds to a core promoter. Proximal promoter elements, these modulate the efficiency of transcription. So we talk about what we say basal levels of transcription. That means, our average levels of transcription. This can modulate it. So proximal promoter elements can make transcription happen faster, or they can make it happen slower. And we'll talk about how in just a moment. In addition to having core and proximal promoters, we can also divide promoters into what their structure and function is. So some promoters are what we call focused promoters, in which transcription, it'll start at the focused promoter and it will give us just one major transcript. This is very similar to what we talked about with the lac operon. That was a focused promoter. It's common in lower eukaryotes. In vertebrates, not as much of our genes are actually focused promoters. We have a lot more dispersed promoters. That being said, we understand focused promoters far better than dispersed, because what dispersed promoters are is there's several start sites. And depending on where the RNA polymerase starts, you're going to get multiple transcripts that can be made. We don't fully understand this. There's a lot of research that needs to be done on dispersed promoters, especially when you consider the fact that they make up 70% of ours, because we're vertebrates, of our genes have these dispersed promoters. What we do know is that they're usually found in those CPG islands, and that they don't have nucleosomes around them, right? Now, since we know and we best understand focus promoters, that's what we're going to talk about for the rest of the lecture today. And I just want to note that this only represents a small portion of the total genes that function this way. So even though it doesn't represent a large number, it's what we understand the best. So let's kind of talk about those. Focus promoters, they're made up of one or more core elements. Okay, so our core element, for instance, could be the initiator. This is where we actually where RNA polymerase starts putting on our RNTPs. The tatabox. We have the TFB2 recognition site. We could have BRE. Right, these are downstream. We can have DPE, which is upstream promoters. These are all examples. The promoters contain then our proximal promoter elements. These are located even further upstream from our tatabox or our BRE core promoters. And these enhance transcription. So if things bind here, we're going to have more transcription occur. An example would be like the cat box, okay, where we have the CAT motif. There's another one called the GC box. Okay, those are promoters. Now, I also mentioned that we have enhancers and silencers. So what's an enhancer? Well, it's a cis-acting site, DNA sequence, and they contain several short DNA sequences that when things are bound to it, they increase transcription rates. So they're located on either side of the gene. They don't have to be right next to it. They can be some distance from a gene, or they can even be inside the gene itself. And if we bind to these enhancers, then we will have maximal transcription. Okay, silencers are kind of the opposite, right? They're cis-acting elements that then repress the level of transcription. So they make transcription more difficult to occur, so we have less transcription. So what's the difference between enhancers, silencers, and promoters? So enhancers and silencers are in the same category. First off, position of an enhancer or a silencer is not fixed to the gene. It can be upstream, downstream, or within the gene, like in the intron. That's not true of promoters. Promoters are always before. Okay, the orientation of an enhancer or a silencer can be inverted without affecting expression. What I mean is that it could be backwards. Promoters, they have to be read 5' to 3', but you could flip an enhancer. You could cut it out, turn it around, stick it back in backwards, and it would still function just the same. And finally, with enhancers and silencers, their function is not gene dependent. What do I mean by that? Well, and if you cut a promoter region out of a specific gene and then you moved it into another gene and replaced, that gene's promoter with that one you cut out, it would not function. We would not be able to transcribe it. So promoters are gene dependent. However, enhancers and silencers aren't. So you can take an enhancer from one gene, stick it into another, and it's going to enhance the transcription of that gene. I've used the term transcription factors a couple times before. These are proteins. They're regulatory proteins, and these proteins target cis-acting sites of genes. So they can bind to enhancers or silencers. So a transcription factor that binds to enhancer would be called an activator, meaning it increases transcription initiation. Transcription factor that were to bind to a silencer or to a promoter for that reason that decreases transcription initiation would be called a repressor. So I don't want to make it sound like transcription factors only bind to enhancers or silencers. They could also bind to promoters. It's just what's their function. If they increase, then they're going to be an activator. If they decrease, they're going to be a repressor. And we talked about this last time with the lac-operon in prokaryotes, right? That repressor protein was a transcription factor, and it bound to the operator. Now, we can take these regulatory proteins and use them, depending on their relative abundance within the cell, to fine-tune whether we're going to transcribe a lot of a particular gene or not very much. If we look at the actual structure of a transcription factor, they have two functional domains. Okay, and what I mean is a cluster of amino acids that has a very specific function. One cluster is going to be what we call a DNA binding domain. So this is going to be the area in which the transcription factor binds to a very specific DNA sequence for which it matches. And it's going to be, generally that sequence is going to be found in that cis-acting regulatory site. It can also have a trans-activating or trans-repression domain. This is the part that activates or represses transcription by binding to not just the DNA, that's the DNA binding zone, but this binds to other proteins, other transcription factors, or in some cases, RNA polymerase itself. what are some of these common DNA binding domains? Well, they create these amino acid sequences create domains like, say, the helix turn helix, or zinc finger, or basic leucine zipper. These are ones we tend to see associated with this. So let's look at what this looks like. Okay, we're going to look at some activators and repressors and how they interact with general transcription factors. And in order to start this, let's just talk about, we're almost taking a step back and talking about how transcription starts altogether. When we talk about transcription, we mentioned how we begin with initiation, then we have elongation, and then finally termination. And that initiation is all about getting RNA polymerase seated on the promoter region so that we can then start transcription. And the process by which we seat RNA polymerase II is not a simple process. It's not just, oh, hey, RNA polymerase II finds that motif and seats itself. No, we have a lot of transcription factors. Your book goes through, this what we call the pre-initiation complex, or the PIC. This is all of the transcription factors that have to be bound in order to hold RNA polymerase II in place. And it begins with transcription factor 2. So transcription factor 2, along with a transcription factor called TBP, binds at the TATA box. Okay, so this comes in. This transcription factor 2A assists. Here we see, our 2D kind of came in, brings all this in. Finally, we get 2B. Lots of transcription factors come in and seat this transcription factor 2D onto it. Okay, this is the 2D. Then we have mediator proteins as well as transcription factors that bind to RNA polymerase. They can then bind down on this PIC, right? And they bind, and when this happens, it basically clamps in and holds this polymerase in place. And what you should imagine is this is causing the DNA to basically almost kind of like bend over in a little bit and create this fully formed PIC that then mediates the unwinding of the promoter DNA and then the elongation to occur. So what happens when we add a transcription factor that binds to an activator or repressor? Well, again, it's going to increase or decrease the rate at which we can start initiation. You saw all those transcription factors trying to bring in the RNA polymerase. That takes time. If you make it more difficult for those things to bind, transcription is going to be a lot slower. If we make it easier, it's going to be a lot faster. So activators and repressors, they can bind to chromatin near the promoter and help open up those regions and make it easier for the transcription to occur. And they can also bind directly to transcription factors to help enhance or inhibit their initiation. So they can bind to the chromatin or to the transcription factors. So let's look at what we call an activator. So here is a cis-acting element. It's an enhancer region on the DNA. And we can see how these activators and co-activator proteins, they can bind on with their DNA binding domain to the enhancer. And then they're also binding onto transcription factors and mediator proteins with their protein binding domains, their transacting domains. And this locks in the polymerase II. What this happens when we have this enhancer thing, RNA polymerase II starts elongating. It moves out. Normally when that happens, all of these transcription factors disassociate and we have to reform the PIC all over again. But with this enhanceosome, it actually retains a lot of these transcription factors in there. It locks them in. So all we have to do is bring in the RNA polymerase II and it can bind very quickly and start elongating again. This increases the number of transcripts that we can get in a given minute, right? Which means we're elevating transcription, we're activating it. Repressor proteins would be something similar, right? Instead of making it easier to hold these things in, they may be making it more difficult. They could bend the DNA in such a fashion to where it makes it difficult for the PIC to form and therefore repress, make it harder for transcription to occur. So that's transcriptional gene regulation. What about post-transcriptional, things that happen after we transcribe our genes. We've actually, believe it or not, talked about quite a few of these, so this should be review. Most post-transcriptional gene regulation occurs at the steps from going from RNA processing to protein modification. So even though we talked about all those transcriptional control mechanisms, probably most of our regulation is actually occurring post-transcriptional, and it plays a role in removing out our introns, splicing exons together. Remember that 5' methylguanosine cap and the poly A tail. Even translation, the ability to translate is going to dictate how many transcripts we have or how much protein we have. And then the stability of those transcripts, we can alter that. This should be review. Splicing, again, alternative splicing we talked about in a previous chapter on translation, on transcription, about how we can splice out exons. And as opposed, that can change. We can get different forms of our mRNAs from an identical pre-mRNA. What that means is the number of proteins that a cell can make, that's what we call its proteome, is not directly related to the number of genes in the genome. We have way more, the proteome is much larger than the genome. In humans, for example, 2/3 of our genes undergo alternative splicing. That's a lot, right? We have several 100,000 different proteins, but there's only 25,000 genes, plus or minus a couple thousand in our genome. So we're making way, way more proteins. If we talk about mRNA stability, each mRNA has a set amount of time in which it is going to be functional within the cell, and then it's going to start degrading. That degradation of the mRNA, we call its half-life. And this determines the amount of mRNA available for translation. So if we increase the half-life of an mRNA, we're going to be able to get more, we're going to be able to translate it more and create more protein. If we decrease that half-life, we won't be able to get as much protein out of that particular mRNA. What are some ways that we can alter mRNA stability? Well, first is we can have enzyme-targeted degradation. You remember that poly A tail has a recognition site for certain enzymes that come in and start cleaving it away. That makes it unstable. The whole function of the poly A tail is to provide stability. But we have enzymes that can target that tail and take it back off. Additionally, we have enzymes that can target the 7-methylguanosine cap on the 5' end, and they can decap the enzyme, or decap the mRNA, and then it gets chewed up. And then finally, the body can produce what we call endonucleases. These are enzymes that can cleave the mRNA from in the middle, right? So they degrade the exposed interior. Even if the ends are protected, if we produce these endonucleases, we break up the middle. A lot of our mRNA stability is going to be regulated by what we call these RNA sequence elements. They help recruit either the degrading or stabilizing complexes. An example of this would be adenosine uracil-rich elements. We call these AREs. We find these in the 3' untranslated regions of the mRNA with very short half-lives. And the reason why they have short half-lives is when we have these regions, they attract, those enzymes bind to them and start cleaving the tail. I'm not going to test you on AREs, I'm just giving an example here. So we talked about mRNA stability. What happens to the mRNA after it then leaves the nucleus and enters into the cytoplasm? It gets translated, and here there's another opportunity for eukaryotic gene expression regulation, in that we can regulate the quantity and activity of a gene product by altering its translation. So we can regulate translation to maybe make it more difficult for these mRNAs to be produced. We could take that post-translational stability of the protein, make that protein unstable so it doesn't fold on its own and therefore maybe is inactive. Or we could even take the protein itself and modify it after translation to change its structure and hence its activity, maybe increase its activity or decrease its activity. All those things play a role in gene regulation. In addition, there's one last thing I want to give a little bit of lip service to. That would be RNA silencing. So the genome can be transcribed to produce short RNA molecules that can regulate our gene expression. And we've talked about some of these when we talk about X inactivation. Remember the XIST transcript? That when it was transcribed would then bind on the XIC locus and basically cause the X chromosome to be inactivated by methylating that chromosome. That's a form of what we call RNA interference. Okay, it leads to RNA-induced gene silencing, and we can use a special type of protein called a dicer. It can take these RNA molecules that we produce and it can cleave them, making them really small, and it can bind to mRNAs and act as what we call small interfering RNAs or sometimes microRNAs. And this represses mRNA's ability to be translated. And in some cases, it can even, when they bind to it, it triggers degradation of that mRNA. And this is a really exciting field in genetics. So in RNA technology, there's been a lot of labs that have been looking about how can we utilize RNA interference to develop a pharmaceutical agent to treat a disease that maybe is caused by overexpression of a specific gene. Or it could be a disease in which we're having just normal expression of an abnormal gene, and that abnormal gene is causing the pathophysiology. Okay, so if they can develop these RNA interference molecules, these microRNAs, and then administer them to people via injection or drug, then potentially we could treat some of these disorders. And we've been doing this for the past couple of decades, getting better at it. There's been promising examples in HIV, polio, influenza, even some myopic disorders with vision, where people's vision has been partially restored by stopping the expression of certain abnormal genes that create abnormal cones in people's eyes. So some really exciting technology plays a role in trying to mitigate this gene expression.